Up first is Tony Vanderpoel speaking about Pacific Oncovirus.
Thank you. It's an honor to be the first speaker here. I guess I have about 60 seconds left to speak. I'm going to talk about a promising oncolytic virus that's composed of genes from Lassa fever virus and from vesicular stomatitis virus. It works very well in the laboratory. We're interested in moving it on to the clinic. It has a number of attributes. First of all, we're trying to target glioblastoma, which is a deadly brain cancer. The virus targets glioblastoma in animal models. Based on the mechanism, it probably will infect many other cancers and kill them. It's a replicating negative strand RNA virus. And unlike many other labs that tend to focus on a single virus, my lab has looked at about 50 different oncolytic viruses over the last 20 years, and this virus appears to be the best of the lot. This is a safety comparison of a virus that's already in clinical trials for liver cancer. The top graph shows survival if we inject that virus. It's a vesicular stomatitis virus that expresses beta interferon. If it goes into the brain, it's deadly. All animals we inject in the brain die. In contrast, when we inject our virus, Lassa VSV, 100% of the animals live. We find no mortality or morbidity in the brain. And that's very important because many viruses are dangerous in the brain. This is perhaps the most important slide in terms of data. If we put human GBMs, glioblastoma, in the mouse brain, the mice die within about 4 weeks. If we give them another virus, which is a Ebola VSV, which is being used in Africa as a vaccine, they live about a week longer. But if we give them Lassa VSV intravenously, so we're not injecting in the tumor, we're injecting into the mouse's vascular system, the mice live indefinitely. Importantly, the virus can move around within the brain. So here we inject Lassa VSV into one of two melanomas. It destroys that melanoma. That's the melanoma on the right, on your right. And then it moves within the brain to the left melanoma and begins to infect that. And that's very important for cancers because cancers are often metastasizing, and that's one of the big problems in how to deal with cancer. How do you deal with the metastasis? This shows that the virus can move around within the body and within the brain to attack metastases. Competition landscape. TVAC was brought forth by Amgen and was the first oncolytic virus approved by the FDA. Amgen paid about $1 billion for that. It's also a virus that you may have seen on 60 Minutes television at Duke University. The poliovirus, if that goes into the vascular system, mice with brain tumors live another couple of weeks. In contrast, when our virus goes into the vascular system, mice live indefinitely. The virus seems to completely destroy the brain tumor. And again, compared with the VSV in clinical trials with interferon, which is deadly in the brain, our virus appears to be safe. So we're interested in taking two steps forward. One is generating a clinical grade virus, which we can't do at Yale. We have to pay a pharmaceutical company to do that. And then testing the clinical grade virus in rodents to make sure it's still efficacious. And finally, testing it in non-human primates to make sure it's safe. After that, we think we can proceed to clinical trials here at Yale. In terms of other oncolytic viruses, there are a number of large purchases being made in the last few years, many of them with amounts approaching $1 billion. One of the smallest was $230 million. So in the last few years, oncolytic viruses have become much more popular than they were before, and a number of large pharmaceutical companies are purchasing the rights for these viruses. We think the Lassa VSV that we work with is safe. It targets melanoma, it targets ovarian cancer, it targets glioblastoma, safe in the brain, and appears to be quite efficacious. Thank you very much.
We have time for questions. Quick questions, somebody. Amanda.
Do the tumors recur in these models?
I'm sorry?
Did the tumors recur?
Go back again in these models?
No, we haven't seen that.
Other questions? Over here.
How do you deliver the virus?
We delivered intravenously, but I think if we were doing it clinically, we'd resect the tumor and then put the virus into the tumor cavity.
Glioblastoma has been known to hide out throughout the rest of the body. Have you found the virus follow it outside the brain?
We haven't done that with glioblastoma. We've done it with melanoma, and if we put melanoma in several places in the body, the virus is able to find the different metastases after intravenous inoculation.
One more quick question? Tony, thank you very much.
Thank you.